Again, welcome in everybody. Thanks for joining us from all around the world. My name is Neela Lay, and I'm the Scientific Marketing Manager here at HighRes. And this is Revolutionizing 384 Well Library Prep, the webinar collaboration between HighRes, Formulatrix, and BlueCatBio. Before we begin, a few housekeeping items. This session will last approximately an hour, and we'll have a dedicated Q&A session at the end, but feel free to submit your questions using the Q&A function throughout the program. This webinar is being recorded, so it'll be shared with all registered attendees by end of day today. All right, let's get started. Today's webinar, like I said earlier, is a collaboration between HighRes and our instrument partners at Formulatrix and BlueCatBio, two companies that are at the forefront of liquid handling technology. And we're excited to bring you insights from our combined expertise in helping research teams streamline workflows, enhance precision, and accelerate discovery through connected systems. We'll go through the challenge and the system first, and then we'll do a deep dive of the Tempest and Blue Washer. And we'll end with a live walkthrough of the protocols in Zelario Scheduler. Hope you're as excited as I am. At HighRes, our work is grounded in a simple but ambitious commitment. We partner with life science organizations to implement intelligent data and lab automation workflows that empower every team member to plan and execute efficient and reproducible science. That commitment comes to life through three things that we hold ourselves to every single day: innovation, accessibility, and reliability. Science doesn't stand still, and neither do we. HighRes combines cutting-edge robotics with intelligent software to fundamentally change how labs operate. From AI-augmented co-pilots that guide scientists through complex workflows to fully autonomous robots that execute them. We're not building the lab of the future, we are delivering it today. Powerful automation should never be out of reach. We believe that regardless of your lab's size, budget, or technical expertise, intelligent workflows should be within your grasp. That's why HighRes offers flexible solutions, from plug-and-play systems to open APIs, so that your team can start small and see results quickly, and scale with confidence. And when it matters most, you need a partner that you can count on. With over 20 years of automation experience, powering high-throughput laboratories, HighRes has built a reputation for uptime, precision, and reproducibility. Our products are engineered to perform and backed by best-in-class customer success teams who are invested in your outcomes. And these aren't just words on a slide. They reflect how we show up for every scientist, every team, and every organization that we serve. Now, let me introduce you to our speakers for today. First, we have Thomas Rollins and Michael Smith from HighRes. Thomas is Vice President of Marketing at HighRes. With more than 12 years of experience in life science product management and marketing, he has helped bring numerous laboratory automation to market, including liquid handling, robotics, and workflow software solutions. Today, Tom will share how advances in automation are enabling high-throughput, cost-effective NGS library preparation at 384 well scale. Michael is applications lead here at HighRes with over 22 years of scientific research and automation experience across biotech and large pharma. He spent 8 years as a primary operator of large-scale integrated HighRes robotic systems before joining the HighRes team, where he has now contributed for 6 years. Mike brings a hands-on technical perspective that is rare in applications roles, grounded in real-world lab operations and deep familiarity with the systems that he supports. Next, we have Claire Duffy from Formulatrix. Claire is a mechanical engineer and product manager for Mantis and Tempest, where she works on precision liquid handling and automation tools. Her background includes product design and system integration, with a focus on making complex systems more practical and reliable. She works to improve day-to-day workflows in research and laboratory settings. Finally, we have Frank Feist from BlueCatBio. Frank is the CEO and co founder of BlueCatBio, which was established in 2014 with a focus on developing innovative liquid handling and centrifugation solutions. Under his leadership, BlueCatBio's products have earned the trust of the majority of top 25 pharma companies as well as leading clinical diagnostics labs, a testament to real world performance and reliability of the technology he helped bring to market. Tom, would you like to give us a brief overview of the system of the hour today?
Thanks so much, Neela, for the awesome introduction. So, I wanted to start out today by discussing with everyone here a framework for NGS library prep. As we look at building an optimal solution that helps scientists achieve their research goals, we feel it's important to put a framework around what it is that it means to actually automate NGS library prep. And the beauty of these workflows is that when you look at the standard operating procedures across DNA sequencing, RNA sequencing, and targeted sequencing processes, the same four building blocks are assembled in different orders to make up all of those standard operating procedures. So it's very well standardized. We have reagent dispensing, which is what I call like a one to many liquid transfer. And that's when we add enzymes, beads, and buffers to samples at low volumes. The second area is bead-based cleanups. This is washing beads and eluting sample from beads as part of our purification process. We have sample transfers, which I call one-to-one transfers. This is when you add UDIs from a UDI plate to a plate of samples directly, and also removing the final eluted sample from a purification plate into a fresh PCR plate. And then the fourth step that makes up these workflows is temperature management. So samples need to be incubated and thermal cycled for reactions to occur appropriately. So with that information in our back pocket, we'll advance to the next slide. And here we look at the different way that these building blocks are executed in labs across the industry. And what we see is that when customers are in 96 well plates, the workflow can be performed in one of three ways. Many labs perform these workflows completely manually. So they're using pipettes to transfer samples, to add enzymes and beads. They're using kind of on deck mixers and shakers to mix tubes and plates. And they're using like a on bench thermal cycler to manage temperatures. In higher throughput environments, we see a semi-automated approach. So that's where users will invest in something like a liquid handler or a dispenser or a plate washer to perform a single step in the workflow, but they'll still manually move plates through the process. So, for instance, we see a lot of customers who have a Formulatrix Tempest and a BlueCatBio blue washer, and they will pass plates between those two instruments into thermal cyclers on the bench and achieve a great deal of the benefits of automation, but without investing in a fully automated platform. And then on the higher end, we see fully automated workflow solutions. So this is where a liquid handler is used to perform all four of the workflow steps that we talked about on the last slide. And this provides the greatest degree of process safety and reproducibility because the samples are only touched twice when they go onto the instrument and when they're being pulled off as high quality libraries at the end of the process. So, like I said, in the world of 96 well library prep, we see all three of these approaches taken. And when we thought as a company at HighRes how we can provide a positive impact and help scientists to achieve their research goals, what we noticed is that when customers move into 384 well plates, the manual approach becomes quite arduous and error prone. And most customers fall into this semi automated bucket where they're using products that facilitate the addition of liquids or cleanups in 384 well plates, but they're still having to move through a manual process step by step. So we thought the biggest contribution that we could have to the market is to look at a gold standard solution for 384 library prep, leveraging our capabilities and experience in automation and bringing together the best products for the job along the way. So with that, I'd ask you to move to the next slide and we can talk about the Prime NGS Workstation. So, in developing this solution, we, like I said, chose the best tools for the job to execute on the framework of tasks that make up the library prep process. For adding reagents, buffers, and beads, we chose the Formulatrix Tempest, and we have our partnership with Formulatrix here. We like this instrument because it allows you to dispense low volumes of liquids, non-contact, and without the use of liquid classes. And um, it also has quite a low dead volume, so it's uh, quite efficient in the way that it processes samples and is automation friendly. For bead-based cleanups, we chose the BlueCatBio Blue Washer. We like this instrument because it allows us to perform what would traditionally take, a handful of boxes of tips to process in a manual or traditional automated workflow, and allows you to perform essentially a consumable-free cleanup. It's also highly automation friendly and quite reproducible and delivers nice results. For incubation and amplification, we chose the Inhico thermal cycler. And then for the one-to-one transfers that I spoke to in the first slide of my presentation, those are the steps that require tip-based liquid handling outright. We leverage our Prime liquid handler to perform those tasks, and we rely on the appliances around the instrument to deliver the um, on the specific tasks that they're designed to perform. Another noteworthy item to mention for the Prime NGS Workstation is that it leverages our Solario software, which is a best-in-class automation software for scheduling plates to move through complex processes in parallel. So with the use of our scheduling software and the tools in this way, we can actually process 8 384-well plates through a DNA sequencing protocol in just 8 hours, leveraging the capabilities of our scheduling software. The last thing I'll mention for the Prime NGS is that with the focus in 384 and the use of the tools that we've chosen, we have actually developed a solution that allows us to miniaturize methods across the NGS library prep world. So we have examples where you can run 4 or 5 times as many samples for the same cost to your lab while achieving a high degree of reproducibility and generate high quality libraries at the end of the day. If you'll move to the next slide, Neela, we'll do a little bit of a deep dive so you can see exactly kind of the Prime liquid handler, which we like to think of as the heart of the system. So the Prime liquid handler is unique in the liquid handling world because it is developed with an automation first mindset. So we know that in high throughput workflows like 384 well library prep, there are a large number of consumables required to execute those workflows. So under the deck of the liquid handler, we have plenty of room to support the pipette tips, plates, and lids that are required to process batches of 8 or more plates through the 384 well library prep process. And the way the Prime is built, the robot that's used to transfer the plates to the deck of the liquid handler for pipetting can also be used to integrate device or lab instruments that sit off the deck. So in the Prime NGS Workstation, we have a table next to the Prime that has all of our third-party instruments on it, and we can load plates to those instruments using the robotic arm as necessary for the workflow. The last thing I'll mention here is the dual deck approach that we have that allows for continuous pipetting. So the way that the Prime liquid handler works is that while one tray of 5 plates is being pipetted on by the pipette head, we are loading a second tray with the robot for the next pipetting step. And then once the pipetting is done with the first tray, we swap the trays and we can load and deal with the automation and movement of the initial plates and continuously pipette and work on the second plate. So this leads us to be able to provide an extremely efficient solution for the pipetting side of the workflow. So with that, on the next slide, we have a brief video so that you can get a sense of the Prime NGS Workstation in action and a bit of a voiceover from our product manager, Hannah.
Hi, my name is Hannah Blakely, and I'm here to talk about the new Prime NGS Workstation from HighRes. It is centered around our compact liquid handler that we make here at HighRes, and also has a bunch of other devices on the back to be able to run an NGS library prep workflow in a 384 well format with 8 plates in an 8-hour workday. First, we'll look at the Prime itself. It is a compact liquid handler that is designed for high throughput and efficiency and flexibility. On the top, we have the pipetting deck. It has 2 dynamic trays and a fixed tray for user access, plus automatically swapping heads. These two dynamic trays are really helping us out in this workflow because there's a bunch of times where we want to be doing a bunch of pipetting back to back. We're doing that with these trays doing tray pooling, which is where one tray is having labware loaded onto it while the other tray is pipetting. This makes it super fast and efficient. In the bottom here, we have ambient storage for all of our labware and our tips for this whole workflow. It could even hold more than what we're using here today. Then on the back, you can see we've got our collaborative robotic arm. It takes the plates to and from the storage and the deck, and also to all the devices that we've got here on the back. And let's give you a brief overview of them. So the Prime itself is the main robot on the system, so we call this a Prime-driven system. You basically need nothing but the Prime and a table with a few devices. On top, we have the Tempest dispenser. It's dispensing a few reagents for us, including the magnetic beads. We also have a BioShake for shaking and our Plate Orient with barcode scanner. On the middle level here, we have the Blue Washer by BlueCatBio. It has been configured for 384 well plates with a magnetic plate on it. And then on the bottom level, all the way down there, we can have up to 4 ODTCs for our thermal cycling steps. Together, all of these devices help us save consumables in terms of not using as many tips, because we're not doing quite as much pipetting on the Prime and we're spreading out the movement and action with all of these other devices. And that's what lets us do all 8 plates on these really long workflows that would take a person about 6 hours to do on their own for 1 plate. We can do 8 plates in 8 hours. So with this powerful workstation, it saves you a bunch of time. Not only is it faster than if a person is doing it manually, but it's doing it all by itself. You just set it up in the morning, forget it, and at the end of the day, you've got libraries ready to go. It also has a cost-saving focus with using fewer consumables and fewer reagents with our dispensing steps. So everything about the system has been designed to save you time and money. Since this has been set up to run with 384 well plates, we have a couple of advantages of that. One, obviously, you get a lot more samples through. And the other one is that our reactions are miniaturized. This means that you're using fewer reagents, which is a lot cheaper. Prime NGS is the most efficient workstation of its kind on the market. If you'd like to learn more, you can visit our website to book a live demo, where we'd be happy to dive into the details of it with you.
So with that, I'm going to hand over to Claire from Formulatrix to do a deep dive about the Tempest.
Thanks, Tom. I'm going to talk about the Tempest, and we can move on to the next slide, which has the introduction to the Tempest one. Yeah. The Tempest is a low-volume, non-contact bulk dispenser that uses our patented microdiaphragm technology to deliver highly reproducible dispensing performance, even at very small volumes. Its broad chemical compatibility is enabled by specialized PFE diaphragm chips, allowing it to handle challenging reagents, including solvents like acetone and DMSO. The system also helps minimize reagent consumption, with just 48 microliters of dead volume per chip. In addition to its precision, Tempest maintains exceptional throughput, dispensing an entire 384 well plate in approximately 5 seconds. Now let's take a closer look at the technology behind Tempest and how those microdiaphragms work. A Tempest can use up to 12 reagents corresponding to 12 chips. Chips are made up of an air layer, which we use to actuate valves and diaphragms, and a liquid layer handling the reagent. System air never touches your reagent. Between uses, chips can be washed and reused with other reagents for thousands of cycles. To dispense, liquid is drawn out of the source container and into the chip. Prime, the reagent is recirculated through the chip and back into the source, which reduces dead volume and prevents settling in the case of beads or cells in suspension. Each chip has 8 nozzles, each with its own cluster of valves and metering diaphragms. To dispense, we pull liquid through an input valve and into one of 2 precisely controlled diaphragms. Dispense volumes are discrete increments of 200 nanoliters or 1 microliter on our low-volume chips and 1 or 5 microliters on our high-volume chips. We then close our input valve and open an output valve, allowing metered reagent to pass through the nozzle and be dispensed. Liquid is dispensed non-contact and has the positional repeatability to be used with labware up to 1536 well plates. So you can see here an animation of the chip dispensing from our large and then our small diaphragm. After a dispense, all of the remaining liquid in the chip and tubing can be recovered, and so that's reducing dead volume as well. Library prep is a laborious part of the NGS workflow. To automate the liquid handling, our instrument must be capable of dispensing low volumes while maintaining high accuracy and precision. It should be flexible enough to be integrated with other automated solutions and should be able to manage high throughput workflows. Tempest qualifies on all four requirements. So let's take a look at those specs for Tempest. Yeah, we can move on. Tempest has a broad volume range, dispensing in increments of 200 nanoliters, 1 microliter, or 5 microliter. On our diaphragm chips and the higher volumes on our continuous flow chips, which can support dispense speeds up to 600 microliters per second. These are also highly repeatable volumes, with CVs under 5% and inaccuracy below 8% at 200 nanoliters. As I mentioned earlier, we have the positional accuracy to support 1536 volt plates, and our recirculate feature ensures uniformity of suspensions. Next slide, please. You can dispense up to 12 reagents at a time through 96 independently controlled nozzles. The Tempest can be equipped with a barcode reader for tracking and can be configured with plate stackers or to be compatible with a robotic arm like it is in this system. It's compatible with high-density plates and can fill plates extremely quickly, depending on the dispense volume. Filling a 96-well plate with 200 nanoliters in around 3 seconds, adding 1 microliter to a 384-well plate in 5 seconds, or 2 mils to a 96 deep well plate in under 2 minutes. Let's go on to the next slide. The Tempest is compatible with a broad range of liquids, including built-in liquid classes supporting reagents up to 20 CP and PFE chips which support harsh chemicals. The system is integration-friendly with networked API support and is compatible with source containers ranging all the way from pipette tips to Nalgene bottles. Continuous flow dispensing allows for accurate, high-volume dispensing. We can go on now. So, why choose the Tempest for NGS workflows? The Tempest can dispense volumes as low as 200 nanoliters with a CV under 5%. It is high throughput and highly configurable. Contamination is virtually eliminated by keeping the liquid path closed and non-contact. And our automated onboard wash also reduces contamination likelihood when you switch between reagents. The Tempest also reduces consumable and reagent waste, which saves on costs. Let's go on to the last slide here. In addition to the sequencing technologies, Tempest is an outstanding bulk dispenser in cell and bead-based assays, PCR workflows, assay development, and compound screening, as well as workflows involving manipulation of nucleic acids like extract purification, CRISPR-Cas editing, and more. If you guys have any questions, I'd be happy to answer them at the end of the webinar. And thank you all.
All right, now let's hear about the blue washer. Frank, the floor is yours.
In addition to the magnetic bead assay application, which is extremely popular for the blue washer is used a lot for cellular assays, in particular for cell-based screening by pharmaceutical companies. And the key advantage there is that it dramatically improves the reliability and predictability of the results from a screen. Just to give you a little bit of background on us as a company, we started BlueCatBio back in 2014. So we're now 12 years in operation, had a fantastic run. We now have an installed base globally of over 750 blue washers in 20 countries. And if I break that down for you, about half of those are standalone desktop machines used by groups of scientists, and the other half is fully integrated. And I would say about 80% of that, of those integrated blue washers have been integrated by HighRes. And it's just a tremendous experience for us working with a team of HighRes, only the best of support. If some issue ever comes up with driver design, they addressed it immediately. And I think all of the blue washers in HighRes installations around the globe are just humming along very happily. So it's just been a really great long-term collaboration. So HighRes has a lot of experience integrating our blue washers for magnetic bead assays, just like in this NGS Prime application. If we advance to the next slide, Nyla. So why centrifugal evacuation for magnetic bead assays? So if we start with what the problems are associated with needle-based aspiration, there's two problems really that stand out. One is the risk of needle aspirating beads away, and the other one is the risk of contamination. Now, of course, both of those have been addressed with conventional needle-based aspiration systems. Most of you are familiar when you have a magnetic bead assay and you're using needle, you have to pull the beads off to the side so there's space for the needle to get to the bottom of the well and aspirate the liquid. Now, that's not a great place for a bead to be when you're trying to wash it, because they tend, to clump together. The surface area of the beads are not really accessible to the wash buffer in the most optimal way. And also you still lose bead loss because of the interaction between the liquid suction from the needle and the beads on the side. The contamination risk in most applications is addressed by the use of single tips, and those, of course, are expensive and they create a lot of waste, none of which you really need. The solution, and Nyla, if you advance to the next slide and start the movie here, the solution is centrifugal evacuation, and this very reductionist video shows how that works. The blue washer essentially is an inverted centrifuge where the plate is positioned inside the rotor in such a way that the wells are facing outwards. So when spinning starts, the G-force pulls the liquid out. So the physics of it are basically exactly the same as what you would do when you do dump and tap in the lab. Nyla, the next slide, please. So the centrifugal evacuation really solves all of the problems I mentioned at the get-go. We have no magnetic bead loss, which of course allows customers to spread their assays or their expensive beads over more assays. It also makes those assays more sensitive. There's no tips. Of course, good for your wallet and also good for the environment. And it stores up space for not having to store that many boxes, like Tom said at the outset. And it's super fast. We evacuate any plate in less than 30 seconds, and that includes the overhead time for opening the door, moving the plate in, spinning, and then opening the door and moving it out again. So when you think of your liquid handler resource or robotic resource, it's very efficient from a cost perspective to offload those tedious evacuation tasks to a blue washer. And the next slide, please, Nyla. So when you saw the video before, how it spins and the liquid comes out, some of you may have asked yourself, well, that looks kind of messy. How do these guys avoid contamination? And, the short answer is now after 12 years of operation, it's really been highly proven by all of our customers, including customers in high throughput human diagnostic applications, that it is just as contamination free as a single tip robot liquid handler would be. And the way we do that is that the initial acceleration of the rotor is very fast. So there's no chance of liquid sort of spilling out gradually and then running over into the next well. When you look at that picture on the bottom right, that's the inside of the rotor enclosure that goes, that basically envelopes the rotor. That metallic sheen is a hydrophobic interior surface, which ensures that the drops, once they splatter against it, move away from it very, very easily. And the mode of action, how these drops are moved away, is the air pressure that's generated by the spinning rotor. So you can think about the rotor spinning, grabbing those droplets, driving them along the interior surface, and then they get removed instantaneously just through gravity via a groove at the bottom of the rotor enclosure. We also have built-in self-cleaning methods that provide additional means to sterilize the interior rotor. Over the years, we've added a lot of functionality on that to also do an immersive deep clean. So we have a whole suite of methods to make sure the instrument is contamination free and stays contamination free through lots of campaigns. And next slide, please. So centrifugal beak separation, how does it work? Basically, you place your microtiter plate on one of our magnetic carriers and let the bead settle. Then you start the blue washer, and the movement expels the supernatant from the plate by centrifugation, while the magnetic beads with the magnets in our carriers retain the magnetic beads at the bottom of the well. And of course, all of that is without contamination. Now, the benefits: no tips, very high throughput, minimal or no bead loss, I would really say. It gives you better data because there is less background and variability, because the amount of residual that you have in your wash buffer is very low, and it's very reproducible, and it optimizes the workload for a liquid handler. The applications are, of course, in the NGS Prime next generation sequencing, library prep, but really any magnetic bead applications such as PCBR cleanup, extraction workflows, or any range of proteomic multiplex bead workflows. On the proteomic side, an additional advantage is that the standard curves look better, which is of course related to the reduction in background. It just gives you more linearity across sample concentrations. And the should I address the question? What was our dispensing? Is it positive displacement? Our dispensing is also a membrane pump, but it's a macro scale standard membrane pump that provides the pressure for the liquid to come out of the nozzle. And now the next page. So that just gives you a sense of what our magnetic carriers look like. It's extremely critical to design the carriers so they have an extremely good fit between the specific plate type that's used and the carrier, because even small gaps between the bottom of the well and the top of the magnet reduce the force with which the magnets are retained. So this is just a small sample of the number of carriers that we have added over the years. So we pretty much have a solution now for most plates out there. And if there's a plate that you guys use that we don't have a carrier for yet, we can always design a custom carrier that eventually becomes part of our product lineup. So that is it. Any questions for the blue washer we'll address in the Q&A section in the back.
Thank you, Frank. Yes, please keep putting in your questions and we will get to those right after this session, or this section of the session. Now we're at the portion of the program where Mike will take us through the protocols. Before we dive into the live demo, he's prepared a few slides for us. So Mike, take it away.
So I just wanted to summarize a few of the key takeaways that Claire and Frank and Tom all mentioned in here. We are doing this preparation in a miniaturized 3D4 well scale, and that allows us to get through 8 3D4 well plates in a little over 8 hours. All these devices that we're putting together on this NGS system, on the Prime NGS system, the Tempest, the Blue Washer, and the Prime, they're all tried and trialed devices in the NGS space. They've been working on standalone, as Frank mentioned. Many BlueCatBio washers are on the on the benchtop as well as Tempests, and our Prime is out there working as a standalone instrument. And we have all these integrated on multiple systems, both large and small. And this is our first offering where we do it in this compact, space-saving footprint, and give you all these capabilities in a small sample size here. So we have the 4 ODTCs at the bottom of the table on the left-hand side of that picture. In the middle, we have the Blue Washer, and the top, the Tempest, connected together with the A cell that's integrated into the back of the Prime. And that allows us to get the storage advantage and store all the consumables for the Prime in the base, and use the tray pooling capabilities of the dual deck on the Prime, as well as the utility tray where we have those magnets as well. Next slide, Neil. So what I'm going to show you today in the protocol design section here is starting with the twist workflow at a 1/5 volume scale, so the low volume scale. And the way that the twist enzymatic fragmentation is separated is in 3 parts if you walk through this protocol. One being the enzymatic fragmentation part, and then the adapter ligation and purification, and then part 3 is the PCR setup and purification of that. And I've tried to maintain the numbering that would match the manuals and the protocol naming as well, so you can kind of follow along if you're familiar with this kit. But that doesn't limit you to just this kit. You can work with your favorite commercial available kit and modify these protocols on your system to meet your needs, because this device suite does match most of the enzymatic fragmentation kits and library prep that are available on the market. One more slide, and then we'll jump to Solario. So to summarize this, I did break this up into 5 distinct protocols. The first one being that enzymatic setup, where we're taking our GNA and putting it into a PCR plate and adding our enzymatic fragmentation reaction mix with the Tempest. And for 8 plates to run through that segment, it's about 2 hours of that initial setup step. And then we're scheduling these back to back. So we go straight from that into the ligation step, where we're taking those DNA tailed fragments, and then we're setting that up for extension, and then purifying that ligating sample using the BlueCat and that magnetic bead cleanup step, and then rolling right from there into the PCR amplification step and number 3, protocol 3 there, where we are taking that ligated adapted GNA sample, PCR amplifying it, and going through one more magnetic bead cleanup step at that 3B section. And that kind of brings you to the final part of the protocol. And with that, each one of these sections, you can see the timing. If we had these all together back to back, they're separated out here. But when you schedule them one after the other, it adds up to about 8 hours. So here's our Solario scheduler. So Solario scheduler is our best in class scheduling software out there on the market right now. We have this organized into 4 separate parts across the top. You have a run and analyze tab, an ordering tab, and a design tab, which we're in right now. So in the designer, we have those 5 protocols already built for you. And if we look at that one at a time, our Solario scheduler software, if you're not as familiar with it, we organize this protocol into what we call threads vertically, going up and down the segments here. So we have a thread for the fragment plate, a thread for your GDNA plate, a thread for your tips, and a thread for our lid steps here. And then as we march down this protocol, this represents what you would do for one set of plates in here. And it's not until the time that you order that you tell a protocol to use, we want 8 plates today, or we want 5 plates today, or we want this particular PCR plate. So this is agnostic to the number or the plastic type that you're using when you're designing these protocols. Our protocols are all drag and drop from operations on this left-hand side here, which we're bringing in line to a thread. We're dragging in the transfer, we're dragging in the dispense, and putting it in the order of your workflow. Each one of these operations that we drop in has a number of devices that are able to do that particular step. So in this case, the transfers are all done on our primes, where we're taking advantage of both the upper and lower tray for tray pooling efficiency as we move along the pipetting events of these transfers. And these flow controls here, we have merges and flow gates are these dotted lines. This allows you to control and keep and maintain your uniformity between plate to plate throughout your assay of the order that you have created. So just in time, we're bringing out the gDNA plate, the fragment plate, and the tips from the storage of the prime, and using that to create the transfer step here on one of those two trays. Then the plate one would move off to the dispenser to get the Tempest to add the enzymatic mix. And then that goes off to the shaker. And the next plate follows sequentially after that. We have a flow gate in here that is uh, equal to the number of the ODTCs that are on the system. So when we do get to that thermocycling steps in here, we have that limited to 4, and that prevents the 5th plate from coming out being set up way in advance and not having a place to go. So these flow controls are in there to prevent backup or uniformity from slipping from plate to plate as you get to that 5th plate and get limited by the number of thermocyclers that are there. You see this mount step. We are taking advantage of a Hamilton Comfort lid here for the PCR lid. So we are mounting that from storage onto our plate right before we go and give that a little shake and put it in the thermocycler. So that's our enzymatic fragment step setup there. If we go and look at the second ligation step here, we are taking those fragments that we've just gone through and tailed, and then we're taking the adapter tips. The adapters on the UDI plate are sitting on our temperature controlled spot on the utility deck of the prime. So we're only moving a box of tips out there with that plate to do that particular transfer from the utility nest. And you can see that we've dismounted that lid to do that. And then we move on and we add in the ligation master mix on the Tempest on the instrument here, and then moving that over to the shaker and back to the thermocycler after we remounted that lid. Now, the next part is the purification part. I think this is the most interesting because as Frank and Tom and Claire had mentioned, we are taking advantage of several features of the instrumentation that are there in order to save you not only volume, but tips and usage here and maintain speed through the protocol process. So when we take the ligated adapters, we're going out to the Tempest to dispense the bead mix, which is recirculating through the bump, which keeping those beads in suspension, getting you uniform bead number dispensed to each one of those wells on the Tempest. We shake that to get that a good mix, and then we incubate that out to let it bind to the particles in there before we take it over to the Blue Cat washer to wash those beads that are now settling on the magnet. And what we do is when we get over there, we give it a little pause before when it sits on the magnet, allow those beads to really settle onto the magnetic plate adapter that's built into the washer. And then we go through those evacuation steps, adding in the wash through the dispenser on the blue washer and removing that dispense, getting that to the point where it's washed and ready to elute. And then for the elution step, somebody asked this question, where are we doing that? We're taking that out back to the prime instrument. We're setting that over to the tray. We match it up in the elution plate. We have a set of tips that comes out there and pulls off your elution to the new plate. And again, we're pausing to allow that plate to sit on the magnet. Any of the movement that might have disrupted those magnetic settling of the beads are now resettled onto the magnet on prime, allowing you to elute your sample without taking over any beads into the next plate. Now, typically, and I'll show you, I have an example on a slide of a non Blue Cat, non Tempest version of this, where we use many more boxes of tips. So I'll show you that in a few minutes as well. And then the last step after that is we are settling in that PCR master mix in here. We're adding our primers, and we're going back to the dispenser to get the master mix on the Tempest, and then we're back to the thermocycler again to go through your final amplification. And then much like the first bead purification, we're going through the purification of that last final sample, where we're coming through, we're dispensing the beads again on the Tempest, We're shaking that to mix that up real good, allow the binding, incubating that to allow that to bind at room temperature, and then washing it again on the blue washer till we get to the final elution and take that sample off, and it's ready to go for your pooling and sequencing needs. So what does this look like when we want to order against this? So Solario has an order tab here, and just move that out of the way. So if we go to the order tab in Solario here, we have the ability to create orders against these protocols that we've created. For your convenience, when we want to add an order, we've created templates for you. So if I want to come in here and make a protocol against my enzymatic fragmentation step here, I have a convenient template built for me, and I can tell it today that it's at this point where I'm going to ask it to do 5 plates today, or I'm going to ask it to do 8 plates today, or I can do 4 plates against the same protocol that's designed here. So I just type in my quantity that I'm running today, recalculate that out, and it knows how many of my other consumables that I need. It also knows exactly where those consumables go in our storage device. For each of these, they have a segment within our Prime store, and then I just place my order against that, and you can see that those have now populated my storage device in my Prime store where those are supposed to be. And there's the tips here, and it's ready to go. I can just save that, come over to my Analyze tab, take a look at what that looks like for today's work. Today I think I said 4 in that one. If I simulate that order against that, we start getting our Gantt charts. And what does that look like for this single first part of the enzymatic fragmentation? I'm going to show you a legend here, and we'll get rid of some lines. So in this particular case, we have our 4 plates that are coming out to do our enzymatic fragmentation in order, maintaining uniformity through pacing and movement with those flow gates. The green step here is our transfer, where we've matched those up, and on Prime we're transferring our gDNA into our fragment plate. And then this little olive bar is our Tempest step, where we're adding our fragmentation. And you can see the notes, the step notes at the bottom of this box that pops up, shows you exactly what the step that I'm following in the protocol from the enzymatic fragmentation 2.0 kit from Twist. And then from there, we go out to a shaker. We've mounted our lid on. Right here at this step, our lid is mounting onto that plate, and then off to the thermocycler number 1 here. And as those dispenses and shakes and building into the thermocycler are happening, we're already got our second plate queued up to pipette onto the next utility tray on Prime, and then so on and so forth for the 4. What does that look like when my resources are utilized? We have another view in here, not just the sample view in chronological order, but we can look at our resource view over time. So here we have our resources and how they're being utilized across there. So you see plate 1 came out to the upper deck of the Prime, the upper tray, and plate 2 was coming out to the second tray. And you can see it's utilizing these tray pooling advantage of Prime that we're continuously loading and pipetting while we're setting up the next deck to be used on the next one. And you can see it goes through the 4 steps of the BioShake and loading up all 4 of those ODTCs. Now, we said we're doing 8 of these plates, so what does that look like when we go through the whole process here? So in this case, I've preset up for the 5 parts of this protocol: 1, 2, 2B, 3, and 3B in order here. And if we want to take a look at what that looks like, we have scheduling tools. So in Solario, we're able to schedule these to take place not only at any particular point in time on a calendar, or we can have it start after a particular order. You can even have advanced scheduling options where you can start it after any protocol thread and any protocol step on any plate number in an order that's queued up before it. So in this case, I'm just taking these one after another and building these back to back with advanced scheduling. So if I were to take this this whole process for 8 plates all the way through and simulate what that looks like, run and give me the same sort of Gantt charts here, where I can see that now I have 8 plates going through this particular enzymatic fragmentation step. And you can see the 5th plate didn't come out until thermocycler 1 is vacated from the first one, and then you start with plate 5, 6, 7, and 8. So you have sort of a staggered delay based on the thermocycler quantity that you have there. And then as you get through to the addition of the tailed fragments out in here, and then through the bead cleanup steps in this particular part here. If I zoom in on some of these, so the first the first cleanup step as we go through, you can see that we're taking advantage of the blue washer's speed as we come through the prime. We've moved that from the tempest over to the blue washer. We're cleaning up that sample, and then we're eluting it out on the prime. So this is what your elution step happens here, and you can see that as the second plate is coming off with the cleanup through the blue washer. We are taking advantage of sequentially running these at the same time. So this is over on the prime, while the second plate is coming in and being cleaned up on the blue washer. So you're overlapping, which is allowing you to get more of these plates going back to back and through the process in a faster manner while maintaining uniformity. We have one more view here. If I look at these plates from time 0, you can see plates 1 through 4 here are treated identically. If we line these all up like they start at the same time, you have no gaps between these, so you get plate-to-plate uniformity throughout your process for all steps of this assay as you move along through the process. Now, I wanted to share one more here because I think the PCR amplification step gives you a good example of the tray pooling. You can see when I zoom in here on the first 2 plates of this process that you are actually still pipetting plate 1 while you're loading up this step over here to the plate 2 to the upper deck of prime. So you're overlapping. So you're not you're taking advantage of the load in, load out time of that deck as it tray pools and moves back and forth. So you got continuous back to back pipetting as your first plate heads out to the tempest for that the enzymatic fragmentation mix. You're not waiting for that to happen here. So it gives you the ability to take advantage of the features of prime while you're moving along through this process. And again, this whole process, when we take it end to end, is 8 hours and a quarter to get through your full 8 samples of 8 3 to 4 well plates of your samples through the whole process. So this is what it looks like when you have on the left-hand side a traditional liquid handler setup of a bead cleanup step versus the right-hand side, which is the one that I was just showing you in the in Solario Live. So you can see on the left that you have many more threads for extra ethanol tips for the washes and your elution setup and your wash tips and your bead addition tips are all being done on the liquid handler side. In this case, you can do that on Prime or any other liquid handler in your lab, but you're managing multiple transfer steps for each and every plate that's going through, which makes your scheduling a little bit more difficult, but it's less cost effective when you use all those tips in addition to that. And you can see on the right, when we do add the tempest for the bead addition and the blue washer for the bead cleanup and the elution just to the prime, you're only using a single box of tips versus 5 on the left hand side, which is giving you time and scheduling efficiencies. And with that, I think I'll leave it for whatever questions came up in the chat.
Thanks so much, Mike. That was incredibly insightful, and the system is clearly so powerful. I'm sure our audience has plenty of questions, and I can already see some coming through. We do have a few minutes. If I can invite all the speakers, our esteemed speakers, to come back on camera to get through some of these questions, that would be awesome. Any ones that we don't get to by the top of the next hour, I'll follow up with an email and some answers from our team. All right? Okay, I'm going to prioritize this first question, and this is for you, Mike. Does the system have a single PC running both Solario and Solution?
Yeah, so the solution software is running in the background in on the same PC as Solara is running. So you got a single PC there, but there is an additional PC that's out on the main platform for controlling some of the other instrumentation too. So if you can use the Solaria and solution PC there, so you have your solution software for making your protocols that we're calling in those transfer steps. So you have all your liquid classes and your pipetting details and your plate setup, just like you would on a standalone Prime. And then we're using the control software, Solaris Scheduler, to coordinate all that happening when you add in the third-party devices.
Awesome. All right. And remember, if you do have to drop at the top of the hour, this webinar will be sent to your email afterwards so you can watch from wherever you left off. All right. Is there a magnet for the 1536 well plate for the blue washer, Frank?
There is not a dedicated 1536 magnet, but the 384 well magnet works excellent with 1536, and we've demonstrated that in a number of applications. Um, so if the person who had that question just email me directly, and I can help you with whatever 1536 magnetic application you have.
Awesome. Next question. Is the system able to pool all samples into one well with single channel capability? Mike?
Yeah, I think that when you get to the pooling step here, a lot of times you need a consumable that can take that in there. We are able to asymmetrically load tips. It will be a little slower on there, but I'm going to throw that one over to Frank because I know Frank's got some solutions for pooling on the Blue Cat, right?
We do, yes. We our new instrument, the Blue Combi X, combines centrifugation and evacuation. It has two doors, and the bottom door works as the centrifuge, and that can be used as a pooling device where we have basically a sandwich of a source plate with, you know, no matter what amount of wells. Like in the NGS, we would use the 384 well with all the libraries in it, and then we flick it over onto the other side and thereby pool all 384 into one cap.
Yeah, so you can do it on the liquid handler in the future. Maybe you do that step on the Blue Cat washer that's capable of pooling. But yeah, that is a pain point and a limitation on some of these assays when you get to that pooling step where you're taking many plates to a single well or many samples to a single well for the sequencing step.
Yes. And I'm unsure of the answer to this, so we'll see who can take this question. But the next question is, um, what step in the process from start to finish limits the throughput?
Yeah, I think you could see that in the Gantt charts a little bit, where you are waiting for that fifth plate at the thermocycler. So that gives you that sort of that step delay to that next spot. But for the most part, you're walking along the process sequentially without waiting. So it really is thermocycler availability that gives you a slight limitation in that assay, but still at 3D4 all samples at 8 plates going through in 8 hours. That's pretty good throughput there.
Awesome. And we'll go through one more question and then we'll have to end the webinar. But the question is, I noticed you had a 96 to 384 reformatting step, step 0. How much time would that add to the overall runtime? And does that storage in the Prime store become limiting? Mike?
Yeah, I think that you can pool those steps. We have these pool by quadrant type protocols that are natively built into Prime. So compressing a 96 well gDNA plate into a 3D4 well portioned out fairly quick. I don't know the timing offhand. I'd have to simulate it there, but it wouldn't add that much more timing for the pre-setup if you're doing that advanced and you have plenty of storage in Prime. And I'll hit that question above it about the temperature control. We do have the ability to put CPACs on the utility deck. So the temperature controlled reagents that are coming from Prime can sit in a temperature controlled plate on the utility deck. And then anything for the Tempest or the reagents that need to be chilled on the outside there, you could use a auxiliary device next to it to keep those things temperature controlled if needed. I think we are out of time.
Yeah, sadly, I want to thank our instrument partners, Frank and Claire, and my colleagues, Mike and Tom, for sharing your expertise with all of us today. That was incredibly valuable. And like I said, you will be receiving a follow-up email with any unanswered questions that I'll gather from the team. And Yeah, if you'd like to continue the conversation or learn more about how HighRes can support your lab automation needs, our contact is here and will be included in that email. Thanks for joining us today, and thanks for all your thoughtful questions and engagement, everybody. We hope you found this valuable and that you can apply these insights to your own lab automation initiatives. Have a great rest of your day, everybody. Take care.
Thanks so much, everybody.
Thank you.